Alan here. This is our first in our series on fats. This one is on insulin resistance. The brain function relies on dietary intake of omega-3 fats to support a broad range of cell membrane properties, particularly in the gray matter, which is rich in membranes. A major structural component of the brain is the omega-3 fat that we call DHA, and it's one of the most important and most abundant fats in the brain. It is an essential nutrition with roles in neurodevelopment, cognition, and in preventing neurodegenerative disorders. This slide is one that we will go over a few times during this talk. Don't be too concerned if we don't cover everything. Generally, this is comparing the omega-3 to the omega-6 fats. We see on the left side, it's the omega-3 fat pathways. On the right side, it's the omega-6 pathways. One of the important things to recognize is the enzymes are used for both pathways. And if you look on the right, the omega-6 goes from the dietary fat, linoleic acid, down the pathway to arachidonic acid, if you find that. And just to the left of there's a pink box that has prostaglandins and other cellular hormones that have an effect on inflammation. You can see that the omega-6 fatty acids tend to stimulate inflammation. And on the left side, we start with the omega-3 fats, that's linoleic acid, and we go down the pathway, and eventually we arrive at DHA, the important omega-3 fat that's used for membranes in the brain. And before we get to there's the EPA fat, which also makes prostaglandins, but these prostaglandins are anti-inflammatory. This is the omega-6 fatty acid chart here. You can see the linoleic is elongated and changed until we get to the arachidonic acid at the bottom. And this is one that can cause inflammation. You can see that enzymes are necessary for these conversion. If you look at this structure, on the left end is the acid end, and on the right end is the carbon hydrogen end, CHO3. And there's what's called double bonds in the middle, and it's called omega-6 because if you start from the carbon end, which is called lipophilic end, you go in 6 carbons, and that's why it's called omega-6 until the double bond. Omega-6 fatty acid is what is called unsaturated, means that some of the hydrogens that are on a saturated fat are not there. They are replaced by having double bonds in the structure. We are at this point going through some of the basic chemistries of fats, so we can discuss it later and we'll have some understanding of what we're discussing. Unsaturated fats. They are less stable and more chemically active than a saturated fat. Here is a triglyceride, 3 fatty acid attached to a glycerol molecule. The first on the top here is what's called a saturated fat. It doesn't have any double bonds. The middle is a monounsaturated fat, means there's only one double bond. And the bottom is an omega-3 fatty acid, and it has a double bond starting at the 3rd carbon. Every time there is a corner in the structure, it is representative of a carbon with hydrogen attached. A triglyceride is the fat that's stored in the fat cells, and they are stored energy, and they can be any type of fatty acid that's attached to these glycerol molecules. In this case, we drew it with 3 different types just for illustration. Plant fat versus animal fats. Animals cannot insert any double bonds before the 7th position. Plants can insert a double bond at the 3 and the 6 position. That's why we call them omega-3 and omega-6. These are called essential fatty acids because we have to get them from a plant source or either from an animal source that originally got them from a plant source. The animals in themselves cannot make these fatty acids and must originally be from a plant source. The idea of competing enzymes for omega-3 and omega-6 fatty acids is an important concept. If you have a diet that's low in omega-6 fatty acids, then you have more enzymes to process the omega-3 fatty acids into the DHA in the brain, and the omega-3 fatty acids are the ones that are low inflammatory and that are needed for the brain structure. So that's one of the reasons why it's good to eat less of the vegetable oils like corn oil, safflower oil, and other refined vegetable oils. This here is a actual more structural model of an omega-3 fatty acid. They are bent and curved, whereas a saturated fat are straight. The saturated fats can pack closer together. That's why they're hard at room temperature, and the omega-3 fats and the omega-6 fatty acids are curved so they don't pack as closely together, and therefore they're liquid at room temperature. The linolenic acid has double bonds at the 3 position, if you count from the first position being at the far right end and count in at 3 bends, which would be the 3rd carbon position. Usually this is a curved shape. However, in the drawing, it's drawn straight. This is the olenic acid. The cis position means that the double bond is on the side that causes a bend. The trans position does not cause a bend or curvature. The normal position is the cis position, which means that this oil would not fit as tightly together and would be liquid at room temperature. Olenic acid is what we call monounsaturated fats, and it's found in almonds and in olive oil and other food products. Some of the nuts have it. The monounsaturated, only one double bond, are more chemically stable than the polyunsaturated fatty acids like the omega-3 or omega-6. They are in olives, nuts, and avocados. This is an actual example showing the hydrogen ions and the carbons and the double bonds in their actual chemical structure. We see that there are 2 hydrogens for each carbon, but if there's a double bond in the chemical structure, there would only be 1 hydrogen. This is the linoleic acid. It is the omega-6 fatty acid, and you can see how it goes down from linoleic acid down the pathway to one of the important things to recognize is the arachidonic acid, which can go into substances that cause increased inflammation. We have a model here of the cell membrane, and it's important when we look at fats to look at the cell membrane because the cell membranes are made of phospholipids, which are fats. The picture of a phospholipid is on the bottom of the picture. It has a hydrophilic head, which means that it likes to be around water, and the lipophilic tail, which means it likes to be in a fatty area. So that's what keeps the cells together, the chemical attraction. The blood is a liquid, so it attracts the hydrophilic, which means water-like, and the other end attracts lipids, so the 2 lipids attract each other and form a layer called a phospholipid bilayer. So this is what the cell membranes are typically made out of. They have protein channels, and they have protein receptors, and all sorts of molecules and receptors in the cell. A single cell is a very complicated structure and machinery, as we can see by this slide. Here we see another picture of the lipophilic bilayer of the cell membrane with the phospholipid model. With the hydrophilic head and the lipophilic tail, one of the lipids in there is straight, representing a saturated fat. The other is bent, which would represent a unsaturated fat. The cell lumen is on one side and the extracellular space would be on the other side. Usually that would have a liquid or could be blood on that side. Interestingly, about the cell wall and the type of fat in the cell wall, the cells have a predominance of saturated fats. They're more tightly and more stiffly packed together. If they have more of an unsaturated fats, they are more mobile and fluid. Here we see the same picture of a triglyceride that we saw before, which had 3 fatty acids. These are usually in fat cells or other places like liver for storage. Saturated fats increase the amount of low-density lipoprotein cholesterol carrying molecules. High levels of LDH cholesterol have been associated with increased risk of cardiovascular disease. Saturated fats are stable at high temperature, unlike polyunsaturated fats. This here is a model of cholesterol. We see they're not at all like the fatty acids that we have seen before. This is a model of a lipoprotein. Lipoprotein comes in several different forms depending on the amount of triglycerides, fats in the molecule and the size. There are high-density lipoproteins, which means they're smaller and more dense. There are low-density lipoproteins that have more triglycerides and are not as dense. And then there's very low-density lipoproteins, and they have quite a bit more fats that they are transporting. And these can be used to assess your risk for atherosclerosis or other diseases. We see in this model that it has the phospholipids covering it, similar to a cell with the cholesterol in the purple inside and the triglycerides in the green inside, and a protein wrapping it around on the outside, which is in the green. This here is a rather complicated model showing the interactions and how the lipoproteins are transported around our system from coming in the liver, the intestines, transported to various organs. Cholesterol is used for various hormone functions, so needs to be transported. Fats cannot be transported in their free state because they are lipophilic. In other words, they wouldn't dissolve in the blood to be transported around, so they need a transport mechanism, and that's what these are. A high-fat diet causes inflammation, and this high-fat diet induces inflammation and is the most important cause of insulin resistance. Insulin resistance is a defect in the cells of our body in response to insulin. The main result is glucose not entering the cell as effectively as desired. The result is an increased blood glucose or sugar and a high insulin level to try to compensate for the high sugar in the blood. Here is a picture of a cell membrane, and we can see in the purple there is glucose entering in through a channel. These channels are responsive and have receptors to insulin, and the channels can be opened or closed, and the blood sugars are regulated by mainly insulin levels. Inflammatory compounds alter insulin receptors by what's called phosphorylation. This reduces the strength of the insulin signal to the cells. This is what is called insulin resistance, that is, reducing the strength of the signal. Here we see an example of a cell membrane. The phosphorylation is there depicted in red. That GLUT4 receptor is affected by insulin and can let the glucose in and out through the channel, regulating the blood sugar or glucose level. Inflammatory mediators that alter insulin receptors and thereby cause insulin resistance include the following inflammatory mediators: cytokine, tumor necrosis factor alpha, TNF, and inflammatory protein kinase such as CJUN, N-terminal kinase, which is abbreviated JNK, and the IIK complex, other cystokines also like IL-1, IL-6, TNF-alpha, etc. So these are inflammatory mediators that alter receptors in the cell. We won't have to remember all the different ones. Mostly from now on, we'll just call them inflammatory mediators, and they could be any of these here. We may just call them cystokines for simplicity. Inflammation, although it is one of the causes of inflammation, has a definite beneficial role. If we are attacked by bacteria or foreign substance, our body needs to have a defense mechanism to fight these off. So the body uses inflammatory mediators to activate our defense system and try to destroy invaders. But sometimes this has untoward effect, as it does with insulin resistance. Another mechanism of insulin resistance is the lipid overload, resulting in a buildup of these DAGs, which is diacetylglycerides or ceramides in the cells. This is a picture of those ceramides in a chemical structure. Omega-6 and saturated fats, especially arachidonic acid and palmitic acid, can be viewed as pro-inflammatory molecules. The common vegetable oils and animal fats are the general sources of these. We see here in this slide, which we've seen before, if you follow down the omega-6 family on the right, you come down to arachidonic acid, which has the pink box, and these are a list of some of the inflammatory molecules that are triggered by the arachidonic acid. And on the other side, the omega-3 family has a similar pathway, but it goes down to EPA, which can activate or form the prostaglandins that are anti-inflammatory and can help reduce the inflammation and cell destructive pathways and can help reduce the level of insulin resistance, since insulin resistance is related to activation of inflammation in the cell's internal structure. Omega-3 fatty acids, especially EPA and DHA, can be viewed as anti-inflammatory molecules. Flax oil, walnuts, cold water fish all have some omega-3 fatty acids in them. The hypothalamus acts to match energy intake to food to calories expenditure to maintain a stable body weight. The hypothalamus is the regulator of our body weight. This here is a picture of where the hypothalamus is, and below the hypothalamus is the pituitary, so there is a direct connection with the pituitary that connects to the body hormones also. Either excess calories or saturated fats, especially palmitic acid, can cause inflammation in the hypothalamus, leading to resistance to satiety. Satiety means having a full feeling or lack of hunger. These feelings are signaled by both insulin and leptin, which are hormones that are produced. As a result of this, satiety is attenuated and hunger increases and weight increases and insulin resistance develops. Here we can see a model of a saturated fat. They have no double bonds and they are solid at room temperature. Examples would be butter, lard. Thus, the presence of adequate levels of omega-3 fats in the diet can decrease inflammation within the hypothalamus. This reduces insulin resistance. Here is a chemical picture of an omega-3 fatty acid. Inflammation increases in the hypothalamus within 24 hours after beginning a high-fat diet, as indicated by increases in the inflammatory mediators. These inflammatory mediators inhibit the normal hormone signaling of leptin and insulin necessary, resulting in hunger. This here is an example of how inflammation works. At the top, we see that injury or infection is present. A cell membrane is disrupted. Phospholipids in the cell membrane are disrupted. Arachidonic acid stimulates the chain of inflammation, including the prostaglandins, leukotrienes, and other inflammatory chemicals, which are a good thing in this case because it's causing resolution of the infection. However, if this is occurring without an infection, it can cause unnecessary inflammation. A high-fat diet can cause inflammation in the hypothalamus, causing inflammatory damage to neurons in the hypothalamus. With time, a high-fat diet decreases the number of neurons responsible for producing satiety signals in the hypothalamus. This here is a picture of neuroimmune crosstalk. The effect of, on one side, a diseased brain by stress, inflammation, and infection can activate the microglia, which are intracellular infection fighters. And we see on the other side, a healthy brain, the microglia are not active and quiescent, not causing any toxic effect to the neuron. So on the right side, it's the inflammatory cytokines and other chemicals that are causing an inflammatory reaction and eventually will damage the neurons. If the diet is rich in palmitic acid, a saturated fat, and it's also produced in the liver by excess dietary glucose, then the hypothalamus pituitary adrenal axis is activated, releasing more cortisol than usual, thereby increasing insulin resistance. If the fatty acid being sensed is primarily oleic acid, such as in olive oil, nuts, and avocados, there will be a reduction in the hormone NPY, a powerful appetite-inducing hormone, which is expressed in the hypothalamus. This decreases hunger. The oleic acid is found in avocados, olives, nuts, chia seed, and other seeds. Here's another picture of the monounsaturated fat oleic acid. We like the cis structure. The trouble with heating, such as frying oleic acid, is it can convert it to the trans, which straightens it out. It no longer has the bend in it, and it acts more like a saturated fat and would be more harmful than it would be if it wasn't heated high. And it would be considered less desirable in this state. The fat cells of the adipose tissue are the only cells in the body that are designed to safely contain large amounts of fat. This is why the adipose tissue is rich in stem cells that can be converted to new fat cells to contain large levels of excess energy in the form of triglycerides. This is a picture of healthy adipose tissue or fat tissue storing lipids intracellularly. As long as those fat cells are healthy, there is no adverse metabolic effect except for excess weight for the person. This is why approximately 1/3 of obese individuals fall into the category of metabolically healthy obese. They have excess body fat but no metabolic disturbance that characterize the manifestations of insulin resistance. Overexpansion of existing fat cells can create hypoxia, which activates the HIF gene. This results in the increase of expression of both the inflammatory mediators JNK and IKK, thereby creating inflammation within the fat cells. This inflammation in turn creates insulin resistance within the fat cells. This here is a picture of an inflamed fat cells where you can see there's a disruption of the fat cells and infiltration with abnormal cells. In adipose tissue, insulin normally decreases the activity of hormone-sensitive lipase. Lipase is an enzyme that breaks down a triglyceride into a fatty acid. We see this here in the model of a triglyceride being broken down into two fatty acids and a monoacetylglycerol. With the development of cellular inflammation and insulin resistance in the fat cells, higher levels of fatty acid can leave the fat cells to enter into the circulation and be taken up by other organs, such as the liver and the skeletal muscle, that are unable to safely store large amounts of fat. This leads to the developing insulin resistance in these organs. Increased inflammation in the fat cells summons an influx of M1 macrophages into the adipose tissue with a corresponding release of inflammatory cytokines, which further increase insulin resistance and release of fatty acids into the blood. We see here a picture of a cell line, and if you follow it on the left side of your screen down the arrows to what you get as a macrophage on the bottom, so you can see the stem cells are more toward the top of the picture. And this shows a picture of the white cell line of blood cells that differentiate into different types as needed. New healthy fat cells could be generated from stem cells within the adipose tissue. However, that process requires the activation of the gene which is inhibited by inflammatory cytokines. However, this gene transcription factor that generates stem cells is increased in the presence of the anti-inflammatory nutrients such as omega-3 fatty acids and plant phenols. Lipotoxicity is the release of large amount of fatty acids from the adipose tissue into the blood. It then accumulates in other tissues such as liver and skeletal muscle. This lipotoxicity increases systemic insulin resistance, and it is with the development of lipotoxicity that the real metabolic consequences of insulin resistance begins. The liver is the central manufacturing plant of the body. Raw materials, primarily fats and carbohydrates, are brought into the body to be processed by the liver and either stored as liver glycogen or reprocessed as newly formed triglycerides in the form of lipoproteins. The liver helps maintain stable glucose levels between meals by balancing the release of glucose versus the storage as glycogen. We see if fat is deposited in the liver, which is fairly common in people with insulin resistance, it eventually deposits, the liver enlarges, scar tissue is formed, and scar tissue eventually becomes cirrhosis and liver damage and failure. The liver cannot safely store excess fat. Therefore, of the first adverse metabolic consequences of insulin resistance is the buildup of fat deposits in the liver. This is known as non-alcoholic fatty liver disease. Currently, about 20 to 30% of Americans have this non-alcoholic fatty liver disease, and 90% of obese type 2 diabetics have non-alcoholic fatty liver disease. It is estimated that 50% of all Americans will have this non-alcoholic fatty liver disease by 2030. Because of the rapid buildup of fatty acids in the liver, the ability to convert them to triglycerides is also overwhelmed, and creamides begin forming in the liver and increasing. The amount of creamides in the liver are the best clinical markers that insulin resistance has begun in the liver. Creamides are also found in other organs when insulin resistance is found and contributes to atherosclerosis. This is a picture of fatty liver cells on the left and a normal liver parenchymal on the right. In the liver, insulin resistance is related only to the fatty acids levels in the liver, not to the level of visceral fat. This may explain why many normal BMI individuals, especially Asians, can have a high level of insulin resistance in the liver. In other words, the Asians may not be overweight, but may have a significant amount of fatty liver if they have had the inflammatory problem that we've discussed. Since the liver also controls cholesterol synthesis, insulin resistance in this organ is reflective of a growing dysfunction in lipoprotein synthesis. This is easily measured by the triglycerides HDL ratio. That is a good clinical marker for liver insulin resistance. A ratio of 3.5 or more provides a simple means for identifying insulin resistance. Skeletal muscles represent a key site for glucose uptake. Cystokines coming from other organs, such as the adipose tissue or the liver, are the main contributors to insulin resistance in the muscles. Creamide levels are strongly related to insulin resistance in the muscles. The skeletal muscles is unique in that exercise can overcome insulin resistance in this organ by increasing the metabolism of accumulated fatty acids and enhancing the transportation of glucose into the cells. This is a slide of the metabolic summary. We see that fats are on the left in yellow, and they're stored in the fatty tissue. The glucose can go down and be stored as fat by being metabolized to triglycerides and stored in the fat by going through the lipoproteins and then transported into the liver. The amino acids can be metabolized, and then by gluconeogenesis they go into the glucose pool in the blood, and the liver and the pancreas and the insulin all regulate the blood glucose levels. Here is a picture of the fatty acid metabolism. We see that the fats can be converted to ketone bodies, and the ketones bodies can be used by muscles or other cells, or they can go into the Krebs cycle where they produce energy. So not only sugar, but ketones can also be metabolized. The ketones are processed and formed from fatty acids in the liver. It should be pointed out that the glycogen storage in muscles can only be used internally as a source of energy and cannot be released back into the circulation to help maintain a stable glucose levels. In other words, the muscles cannot release glucose. The release of glucose must come from the liver. We're going to discuss briefly on the pancreas. The beta cells, which make insulin in the pancreas, are sensitive to glucose levels in the blood and secreted insulin in response to those levels. Beta cells are very prone to toxic mediation by inflammatory agents. In particular, the 12-HETE derived from arachidonic acid is very toxic to the beta cells. The destruction of beta cells by 12-HETE, the pancreas is no longer able to maintain compensatory levels of insulin secretion to reduce blood glucose levels, and the development of type 2 diabetes is rapid. Arachidonic acid is from the omega-6 pathway, another reason to reduce the amount of these vegetable oils. We see here on this chart again that the omega-6 fatty acids go down to the arachidonic acid, which can produce this 12-HETE, damaging the pancreas and sending the person into diabetes. The type of bacteria in the intestinal tract are different in lean and obese individuals. The microbial composition may be a source of low-grade intestinal inflammation via endotoxin mediation by the lipopolysaccharides, LPS, component of gram-negative bacteria. In other words, abnormal bacteria in the intestinal tract, especially the colon, causes inflammation. We have pictures of these LPS found on the walls of the gram-negative bacteria that can be released into the intestinal tract and then migrate into the blood circulation, causing increased inflammation, which would contribute to insulin resistance, since inflammation is the main cause of insulin resistance. A single high-fat diet or high-carbohydrate meal can induce such endotoxemia because of the increased permeability of the gut during digestion of a high-fat meal. Thus, a diet that is high in protein and low in both carbohydrates and fat should reduce the endotoxemia and then decrease inflammation and insulin resistance. It has also been recently demonstrated that composition of the high-fat diet, either rich in saturated fats or other fats, can dramatically alter the composition of the gut microbe and influence the level of endotoxemia in animal models. So just in summary, a high-fat diet overloads the adipose tissue storage capacity and causes insulin resistance, being increased inflammation by various methods discussed. Saturated fats are the most harmful. Omega-6 fats increase inflammation. Omega-3 fats decrease inflammation. Monosaturated fats are not inflammatory. They decrease appetite and are healthier fats than omega-6 fats or saturated fats, but add to the total fat burden in someone with insulin resistance. High temperatures damage omega-6, omega-3, and monounsaturated fats, making them inflammatory. So, recommendations in summary is to avoid saturated fats, lard, butter, cream, coconut oil, palm oil, beef, and animal fats. Use eggs sparingly because eggs have saturated fats, but they do also have some omega-3 fats, which are beneficial. Avoid frying all oils, but especially vegetable oil. High heat changes the structure of oil, making it harmful and inflammatory. Avoid all refined oils such as canola oil, corn oil, soybean oil, safflower oil, palm oil, sunflower oil. Excess omega-3 oils is inflammatory. Include some monounsaturated fats in your diet in the form of avocados, almonds, other nuts, and olives. Include some omega-3 fats in your diet in the form of flaxseed, walnuts, chia seed, and fish like salmon and anchovies. Eat a low-fat diet with a total fat in the diet of 20% or less of total calories.